Okay, great. So we just covered retrovirus multiplication cycle. So here's a HIV multiplication cycle. So it's kind of nice that HIV is basically a retrovirus. So most of this will be the same. So I'll just walk through everything and just really focus more on the differences in here, which is really nice. So far we've seen that rhabdoviridae and paramyxoviridae, they share the same multiplication cycle for the most part, and we saw the same thing for polyomaviridae and papillomaviridae, and we see the same thing for retroviruses and HIV. So as always, begin with the virion, and you attach. Right, you attach using GP120. That GP120 actually goes just like in RSV, it undergoes like a slight change in shape to expose the transmembrane protein, which is smaller than the surface protein. You get fusion. Instead of LDL receptors, though, it uses the immunodeficiency retroviruses. They attach to the T cells, usually of mammals, from what I understand. These things only infect mammals, and they infect CD4, and they don't just infect CD4. They don't just attach to CD4. They also attach to a co-receptor. So you can see here, co-receptor is CCR5 and CXCR4. I'm not going to remember CXCR4, to be honest. I'm just going to remember CCR5. It's just the way my brain works. Essentially, when you finally attach two of those receptors, so one CD4, one CCR5, you get GP41. You get fusion. So here's a fusion, uncoats, same thing, reverse transcriptase with the RNase. Okay, make the double-stranded DNA. HIV does not carry an oncogene. So unlike the retroviruses, some retroviruses don't have the oncogene, but they can still cause cancer by insertional immunogenesis. Others do have the oncogene, and they can cause cancer because they're oncogene. These immunodeficiency viruses, though, simian immunodeficiency virus, feline immunodeficiency virus, human immunodeficiency virus, they attack T cells and they kill the T cells. They don't turn the T cells cancer, you just outright kill it. So you have the double-stranded DNA, and it goes in to nucleus, again for the same reasons as retroviruses. So only in the nucleus can more of the double-stranded DNA be replicated and so forth. Only in the nucleus can you convert from double-stranded DNA into mRNA. You also have splicing the mRNA. Sometimes you don't have splicing if you want to get the GAG and the PAW genes translated. And then if you need your envelope genes, and you do a single splice of the mRNA. And then so we'll cover those. So here's the full-length transcript. Again, same thing as retrovirus. You translate the full transcript, mRNA transcript, without a frame shift. You get the GAG gene. You translate the whole thing with the whole mRNA with a frame shift, then you get the PAW. Proteolysis, essentially protein maturation, with protease being the protein that helps mature these GAG and PAW proteins. So they go there. Your envelope proteins, so these encode your glycoproteins in your envelope. Okay, go there. Simple. What makes HIV special and different is the fact that it has 6 regulatory proteins that retroviruses don't usually have. These regulatory proteins are essential for the pathogenesis of HIV. So VIF right here, for example, HIV needs VIF because without VIF, HIV would get destroyed by a cellular protein called APOBAC. APOBAC basically messes up, it deaminates, it messes up the viral genome, so that when the virus buds out with that messed up genome, its genome so messed up that it can't even infect new cells anymore. VPR stands, I should just go VPU. VPU stands for viral protein unique to HIV. This one destroys CD4 and tetherin. So, you know, see, actually, look at that. All that, look at that. So, you know, VPU inhibits tetherin. They don't show that it inhibits CD4, but it does. You know, you destroy the CD4, now this particular cell can't let the immune system know that something's wrong with it. The tetherin, by inhibiting tetherin, you no longer allow the cell to withhold the viral molecules inside of it. I go into more detail about that in the lecture video that I made earlier today for HIV. SAM HD21 basically, it tricks the cell into thinking that that everything is going fine and dandy, I believe. You have like these TAT and REV. Essentially, TAT makes it so that you get better, more efficient mRNA transcription. REV exports the mRNA transcript out of the nucleus. NEF basically, if I remember, NEF just basically tells the cell to, NEF is a trap. It's a bait that's made by the HIV. So it tells the T cell that's infecting to not kill itself. And it also acts as an attractant for other T cells to come, so that when those other T cells come, the T cell that's infected is budding out a bunch of HIV. And now there's a whole cluster of T cells outside of this, this dirty infected T cell, and that HIV can infect those new T cells that came to the site. VPR, I can't really remember what that is. I almost feel like it's at the tip of my tongue. But moving on, essentially these regulatory proteins right here, they go back and they mess stuff up for a cell. It basically drives the infection forward. That's why you see when they are translated, they go back this way, right? So, you might have like basal levels of translation and transcription, but once, so you get like this tiny amount of regulatory proteins, but once these go inside back into the nucleus, now the virus can really copy itself and make more of itself. So it goes back in here and you get more splicing, more export of the mRNAs, more mRNAs, more viral proteins, and then you can take the full-length transcripts along with the proteins to get particle assembly. So that's HIV, extremely similar to RSV. Okay, now we're going to move on to the next virus.